The overall goal of the single sensillum recording is to investigate neuronal responses of the antennal olfactory sensillum to various odorants.
This method can help to answer the key questions in the field of chemical ecology, such as if insects can detect the chemical odorants from their environment and how they process the chemical odorants. The main advantage of this technique is that it allows measuring the single neuron activity and function. It provides more precise measurement of the sensitivity of individual neurons to the chemical odorants.
The implication of this technique extends toward the development of promising attractants or repellents in the insect control, because this technique can be used to screen large amounts of semiochemicals that are biologically important for the insect.
Begin this procedure by anesthetizing bedbugs on ice for 2 to 3 minutes. Then, fix the insect's antennae and body on a microscope cover slip with double-sided tape, and remove the legs with fine scissors. Use a small pin to gently adjust the antennae on the tape. Next, rest the cover slip against a small ball of dental wax to facilitate manipulation, and adjust it to a 90-degree angle for placing the recording electrode. Once secured, place the bedbug under a stereo microscope. Turn on the light source and adjust the intensity of illumination until the antenna is clearly presented. Then, focus the microscope on the second flagellum of the bedbug's antenna at high magnification.